Good evening, everyone. This is Sailaja from MSc second semester Microbiology. So today my topic is human infecting viruses. So coming to human infecting viruses, about 270 viruses are known to infect humans. Some of these viruses have been known for centuries. Sir, first slide only, sir. So whereas others have recently emerged. Coming to table of contents, the introduction and classification of viruses and barriers to cause infections, diagnostic methods in virology and treatment comes under antiviral drugs, and conclusion. Introduction. The term virus was coined by the Dutch microbiologist Martinus B. Genrich. He was born in 1898, was the first to call virus the incident of the tobacco mosavirus. He showed that the incident was able to migrate in agar gel, therefore being an infectious soluble agent or contagium vivum fluidum, and definitively not a contagium fixum as would be a bacteria. So Jamil, he was born on August 8, 1957. He is an Indian virologist, and he was formerly head of the scientific advisory group to the Indian SARS-CoV-2 genomics, and he established in 2020 December, and he is known for his research in hepatitis E virus. So he elected a fellow of all three major Indian science academics, that is the National Academy of Sciences India, Indian Academy of Sciences, and Indian National Science Academy. So he awarded him the Shanti Swarup Bharat Bhatnagar Prize for Science and Technology. Definition: viruses are obligate parasites, so they can only reproduce inside a living host. A virus is an infectious microbe consisting of a segment of nucleic acid, either DNA or RNA, surrounded by a protein coat. Virus cannot be replicated alone. Instead, it must infect cells and use components of the host cells to make copies of itself. So these are the types of the viruses. Coming to animal viruses. So we all know animal viruses mean these are obligate parasites that infect animals. Their genetic material is either DNA or RNA, which can be single or double-stranded. So they are known to cause serious human diseases such as AIDS, rabies, smallpox, herpes, polio, hepatitis A, and many more. And coming to plant viruses are intracellular parasites that infect plants and cause diseases. So their genetic material is mostly RNA that can be single or double-stranded, and most plant viruses are rod-shaped. So these viruses do not have the machinery to replicate without the host organism, and they do not have envelopes around them. Tobacco mosaic virus is a plant virus and was first to be covered. And second thing, bacteriophages. So these bacteriophages are known as phages, are viruses that infect and replicate only in bacterial cells. So they are ubiquitous in the environment and recognized as the most abundant biological agent on the earth. So these phases size ranges from 24 to 200 nanometers in length, and these are included with two types of the bacteriophages. One is lytic bacteriophages and temperate bacteriophages. Coming to types of viruses. So there are so many types of viruses are there. So based on their structure, and helical viruses like tobacco mosaic viruses, which infects a number of different types of plants, have a slinky shaped capsule that twists around and encloses it genetic material. Coming to polyhedral viruses like an adenoviruses, which are known to cause a range of illness from pink eye to pneumonia are composed of genetic material surrounded by a many-sided capsid, usually with 20 triangular faces. Spherical viruses like infamous coronavirus are essentially helical viruses enclosed in a membrane known as envelope, which is spiked with sugary proteins that against in striking to and entering host cells. And complex viruses are bacteriophages, so which infect and kill bacteria, and a lunar lander and are composed of a polyhedral head and helical body, that is the tail sheath, and legs that attach to a cell membrane so that it can transfer its genetic material to the host. And human immunodeficiency viruses, HIV virus, first one is, and tobacco mosaic viruses, size ranges from 20 nanometers, and adenoviruses in 50 nanometers range, and influenza virus also 50 nanometers, and 80 to 200 nanometers in diameter. Next slide, sir. So classification of viruses on the basis of genome. There are two types of classification based on genome. One is DNA viruses and second one is RNA viruses. Coming to DNA viruses, genome is dsDNA and ssDNA. And dsDNA is categorized into non-enveloped and enveloped. So comes under non-enveloped includes the adenoviridae and papillomaviridae. So in adenoviridae, common cold and viral meningitis, and papillomaviridae, cervical cancer comes, and in enveloped includes the poxviridae and hepsviridae. So it is enveloped that replicates through an RNA intermediate. It is hepatinviridae, that is hepatitis B. And coming to ssDNA, it is non-enveloped DNA virus, includes parvoviridae, that is gastroenteritis. And in RNA viruses, genome is divided into ssRNA and dsRNA. In ssRNA, it is a positive strand and categorized into negative strand. In positive strand, there are two types. So one is non-enveloped as usual and enveloped also. So in non-enveloped positive strand RNA viruses includes picronaviridae. Examples are polio and hepatitis A and gastroenteritis. And in enveloped RNA viruses, there are flaviviridae and coronaviridae, retroviridae, that is SARS virus, and for coming under retroviridae AIDS. And in DSR RNA is a non-enveloped, includes the reoviridae, so that is example of gastroenteritis. These are the human infecting diseases caused by the microorganisms. So bacterial meningitis is caused by the Streptococcus pneumonia and Neisseria meningitis and Streptococcus agalactiae. And skin infections are caused by the Staphylococcus aureus and Streptococcus pyogenes and Pseudomonas aeruginosa. And pneumonia is caused by the community acquired. These are categorized into three types. One is community acquired, atypical, and tuberculosis. So community acquired disease microorganisms are Streptococcus pneumoniae and Staphylococcus aureus, and atypical Mycoplasma pneumoniae and Chlamydia pneumonia. So sexually transmitted diseases caused by the Chlamydia trachomatis and Neisseria gonorrhoeae, and eye infections are caused by the Staphylococcus aureus, Neisseria gonorrhoeae, and sinusitis. These are caused by the Streptococcus pneumoniae, Haemophilus influenza, and gastritis is caused by the Helicobacter pylori, and food poisoning are caused by the Campylobacter jejuni and Salmonella, Shigella, Clostridium, and Staphylococcus aureus and E. coli. And urinary tract infections are caused by the E. coli and other Enterobacteriaceae, Staphylococcus, and Pseudomonas aeruginosa. So These are the infecting viruses. This is the Ebola, and Ebola is caused by the infection with a virus of the family Flavoviridae, and genus is Ebola virus. So, a virus that can cause severe bleeding, organ failure, and that can lead to death. Also, humans may spread the virus to the humans through contact with body fluids such as blood. And initial symptoms include fever, headache, pain, and chills. Later, a person may experience internal bleeding resulting in vomiting or coughing blood. So, route of the transmission is Ebola is spread by direct contact with blood or other fluids, other body fluids such as vomit, diarrhea, breast milk, sweat of infected person who has symptoms of Ebola. Next one, pneumonia. It is caused by the Streptococcus pneumoniae, and causes of pneumonia: influenza viruses, respiratory syncytial virus, and SARS-CoV-2. This is the one of the virus which we can see in COVID-19 symptom also. So, four stages of pneumonia, that is congestion, red hepatization, and gray hepatization, and resolution. So, some of the symptoms are systemic, comes under systemic high fever and chills and lungs. Particularly in pneumonia, we can see lung infection. So, we can't able to take breath, and a shortage of breath, and chest pain, muscle pain, and as usual heart rate, and high heart rate, and some sort of mood swings and joint pains too. So poliomyelitis is a highly infectious viral disease which mainly affects young children. It is transmitted through food and water and multiplies in the intestine, from where it can be invade the nervous system. So healthcare officials recommended that 4 polio shots in childhood is must and should. So first shot at 2 months old, and second dose at 4 months old, and third will be at 6 to 18 months, and booster shot will be between 4 to 66 years old. So there are two types of polio vaccines. One is live attenuated, it is oral vaccine, and inactivated. So the inactivated polio vaccine is contains polio virus that being treated. It can multiply anymore, so it can't multiply anymore. It has an inactive version of polio types 1, 2, and 3. It will give in the form of injections and oral polio vaccines also. So OPV contain 1 or 2 or 3 types of polio viruses: monovalent, bivalent, and trivalent vaccines. Coming to influenza viruses, these are the family member of Orthomyxoviridae. This family represents enveloped viruses of the genome, which consist of segmented negative single strand RNA segments. These are three types of influenza virus. One is influenza A and B and C. So type A and subtypes of type A determined by hemagglutinin and neuraminidase. So type A, it is moderate to severe illness, and it affected all age groups, and humans and other animals are mostly affected. And type B is mild disease, primarily affected in children and humans only. And type C, rarely reported in humans, caused when they breathe in teeny airborne droplets from the cough or sneezes of the someone who has flu. So COVID-19 symptoms compared to flu, almost COVID-19 symptoms and flu similar. Has similar symptoms in fever, dry cough, shortness of breath, and headache, body pain, muscle pain, sore throat, and fatigue, diarrhea, running nose, sneezing, almost similar to COVID-19. And the H1N1 flu, sometimes called swine flu, it is a type of influenza A virus. During the 2009 to 10 flu season, a new H1N1 virus began causing illness in humans. It has often called swine flu and was a new combination of influenza viruses that infect pigs, birds, and humans. Coming to HIV and AIDS, all we know that HIV is a human immunodeficiency virus. HIV and AIDS is acquired immunodeficiency syndrome. So after HIV, the final stage is AIDS. We can tell that. So HIV finds and destroys a types of white blood cells, that is T cells or CD4 cells that immune system must have to fight disease. And in AIDS, the final stage of HIV infection, when someone has one or more specific infections, certain cancers or a very low number of T cells, he or she is considered have AIDS. So the most common methods of transmission of HIV is unprotected sex, that is sexually transmitted disease, HIV and AIDS, we know, and sharing the needles with infected person, and almost eliminated as risk factors for HIV transmission are transmission from infected mother to fetus and infection from blood products. So diagnosis of HIV are and AIDS, types of laboratory HIV test. It is EIA test, a test that screens for the presence of HIV antibodies in blood. And second one is Western blot test, a test that detects HIV antibodies and confirms the results of earlier EIA test. It is CD4 may be run when the Western blot test comes back positive, and RNA viral load test shows how many copies of viruses are circulating in blood. And in CD4 looks at the number of WBC, I mean white blood cells in a sample of blood. And rapid test also, it can give the results, that is HIV test that produces results in 20 minutes. In hepatitis viruses type, name of the virus, these are 5 types of the hepatitis. So hepatitis A, B and C and D and E. So classification, hepatitis E classification comes under Picronaviridae, coronavirus, and the viral genome is ssRNA, and transmission is enteric route. Incubation period of hepatitis A virus is 15 to 45 days. And chronic hepatitis, no. And there is no cure to hepatitis A virus also. And coming to hepatitis B virus, it is hebdanovirus, and transmission of hepatitis B is parenteral, and incubation is 45 to 160 days. And chronic hepatitis, yeah, there is 10% of chances to um, to get chronic hepatitis. And hepatitis C, it is a flavivirus, and also it is hepatitis C and D, also parenteral route. And incubation period of C is 15 to 1 days, and incubation period of hepatitis D is 30 to 60 days. And here in hepatitis C, 50 great than, I mean, more than 50% of the chances to lead chronic hepatitis. And there is no cure to hepatitis B, C, and D, as usual E also. But we can treat with alpha interferons and tackle the symptoms. And measles and chickenpox, coming to measles, acute contagious viral disease characterized by the eruption of red spot on the human skin, and fever is about 40 degrees centigrade. So the common symptoms are cough and conjunctivitis and coryza. So caused by the rubeola virus, which develops in mucus of nose and throat of a infected person. Measles infection in a pregnant woman may result in birth defects, and can even lead to miscarriages also sometimes. So chickenpox is a highly contagious disease caused by the Varicella zoster virus, and it can cause a blister-like rashes among other symptoms. And first rashes appear on the chest, back, and faces, and then spreads over the entire body. So chickenpox illness usually lasts about 4 to 7 days, and then rashes that turns into itchy and fluid filled with blisters. It like so spreads very easily by touching chickenpox blisters or through the air. When someone with chickenpox, so if he or she coughs or sneezes, obviously chickenpox will transmit to one to another. These are symptoms of chickenpox and measles. Similar symptoms we can see here in fever in headache and blisters um, filled with and running nose, body ache, and spots. Itchy blisters appears on skin. Coming to Hep simplex virus, this is also called as a HSV, Hep simplex virus infection. So, coming to family Heps viridae, a virus causing contagious sores most often around the mouth or on the genitals. So, these are the two types of the HSV. So, one is a cold sores infection with Hep simplex virus around the border of the lips, and genital Heps, common sexually transmitted infection marked by the genital pain or sores. So, the average incubation period for initial Herpes infection is 4 days after exposure. So the vesicle break and leave painful ulcers that may take 2 to 4 weeks to heal after the initial Herpes infection. Coming to rabies virus, so we all know that rabies is caused by dog bite. Just give me a second. An acute neurological disease caused by virus rhabdoviridae that attacks the CNS. So it causes the altering symptoms of depression and agitation, and almost invariably leads to extreme type of violent behavior and death. So immunofluorescent antibody test is the best method for rapid diagnosis test for rabies. So rabies vaccine also there developed by the Louis Pasteur in 1885. It is a freeze-dried or liquid preparation of suitable approved strains of fixed rabies virus grown in a approved cell culture or embryo duck or chicken and inactivated by the validation method. So the freeze-dried vaccine is reconstituted immediately before use as a stated in the label to give a clear, slightly opalescent solution or a suspension. So we know that rabies virus is transmitted through direct contact, such as through broken skin or mucous membranes in the eyes, nose, or mouth, with saliva or brain, nervous system tissue from infected animal. People usually get rabies from the bite of a rabid animal. And yeah, and the incubation period of rabies is about 20 to 90 days, and it immediately affects on the central nervous system and some of the salivary glands. And yeah, so coming to coronavirus, we know how we affected with corona COVID-19. So it is caused by the SARS-CoV-2 virus, and COVID-19 can cause mild to severe respiratory illness, including death. The best preventive measures include getting vaccinated, wearing a mask during times of high transmission, staying 6 feet apart, and washing hands often, and avoiding sick people. So there are two kinds of coronaviruses, and one is SARS-CoV COVID and MERS. So severe acute respiratory syndrome coronavirus and Middle East respiratory syndrome coronavirus, which have been in a global outbreak. So coronaviruses enveloped positive sense RNA viruses are characterized by club-like spikes that project from their surface and an unusually large RNA genome and an unique replication strategy. So So COVID-19 is a beta coronavirus in the same subgenus as the severe acute respiratory syndrome, that is SARS virus, as well as several bad coronaviruses, but in a different clade. So for transmission of a coronavirus to occur, a virus must enter a host through a portal of entry, replicate or disseminate within the host, and can be transmitted to a new host through portal of exit. So it is commonly the incubation period of COVID-19 is 5 days, and in addition, about 97% of people who can contact the virus will show symptoms within 11 days. So that means most people who have been infected with the novel coronaviruses will likely show symptoms within 11 days.
Studies show that studies show that
So how do viruses cause disease? By three stages. One is by damaging or killing the cells outright. So by inducing immunopathology, by transforming cells, leads to cancer. So non-immunological responses, phagocytosis, body temperature, hormones, and malnutrition, and age. I mean, it infected based on age also.
yeah, full stop, full
So
stop. That.
by damaging cells or killing cells, mean example polio virus. It is cytolytic and destroys
Sorry,
motor neurons in the spinal cord, and so cause paralysis also
continue,
sometimes.
madam.
Not sometimes, in every time it cause paralysis.
Immediate.
And barriers to the infection, inherent barriers, skin and lack of membrane receptors. So inherent barriers, the host has number of barriers to infection that are inherent to the organism. So these represent the first of defenses which function to prevent or limit infection. And in coming to skin, the skin acts as a formidable barrier to most viruses, and only after this barrier is breached will viruses be able to infect the host. And in lack of membrane receptors, viruses gain entry into the host cells by first binding to the specific receptors on the cells. In the immune system, yeah, antibodies bind to the antigens on the surfaces of target cells and then making them for destruction through the several immune pathways. One is antibody dependent cell mediated cytotoxicity, and then it will leads to phagocytosis and ingestion of the targeted cells, and then complement dependent cytotoxicity. So how host responds in three ways. One is non-specific and immunity humoral and cell mediated. So specific immune response to the viruses is mediated by the combination of humoral and cell-mediated immune mechanism. So cell-mediated immune response, most important in host defense. One a viral infection is established. So CD8 plus T cells, that is cytotoxic T lymphocytes, and CD4 cells, that means helper Th1, that is helpers lymphocytes, T lymphocytes, are then main components of the cells mediated antiviral defense. And coming to the humoral immunoglobulins, that is IgG and IgM and immunoglobulin A. Immunoglobulin A is a mucosal surface secretory immunoglobulins, and antibodies neutralize with the help of complement. Yeah, as usual, HIV destroys the CD4, then
One, two, three.
the infected cells can transmit to the host, then by destroying the CD4 cells. And evasion of immune mechanism by viruses. Viruses can also escape immune attack by changing their antigens. A large number of viruses evade the immune responses by causing generalized immunosuppression. So, by using interferons, uh, it is a protection against viral infection, contain host-coded proteins produced by viral and non-viral inducer on exposure to interferon cell, produces translation inhibiting proteins. TIP inhibits the translation of mRNA, but no effect on cell mRNA viral transcription inhibited. And so inclusion bodies also there. So inclusion bodies are nuclear or cytoplasmic aggregates of stainable substances, usually proteins. They typically represent signs of viral multiplication in a bacterium or eukaryotic cells. and usually consists of viral capsid proteins.
Shailaja,
Shailaja,
Sir.
we need to wind up fast
we need
So
to wind up
diagnostic.
first because core
Yes,
semester
sir.
students are waiting.
Yes, sir. So diagnostic methods in virology, there are a direct examination, indirect examination, and serology. So in direct examination, antigen detection, light microscopy, electron microscopy, and viral genome detection. And indirect examination, cell culture, eggs, and animals are there. By these, indirect examination can be done. And in serology, RIA and EIA test can be done. So, and these are the antiviral drugs we can use to treat the viral infections. So Heps virus is virus polymerase is targeted. So by using acyclovir and we can treat it. And retroviruses, that is HIV viruses, we can use gidovidin and lamivudine and ritonavir and indinavir, we can use it. So by targeting HIV protease and reverse transcriptase, we can treat it. By using hemagglutinin protein and a neurominidase inhibitor, we can treat influenza A and B also. So coming to conclusion, they form part of life. Viruses are used for body to treat illness and sometimes deliver vaccines and diagnose infections. They are used to research tools in illuminate biology and disease and develop the new drugs as usual.
Thank you.
Thank you so much for giving this great opportunity. Thank you, sir.